Hi, let's revisit the topic of molecular evolution in this second video and also revisit a couple of topics such as homology, orthologs, and paralogs. So what is molecular evolution? It's just evolution that occurs at a molecular level. So it's a level that we observe in DNA, RNA, and proteins. It's that process. It's also the science. It's the science of studying the process of that molecular evolution. It's kind of like MSA that way. We refer to both the process and the result, or in this case, the science. And in particular, we're going to be inferring evolutionary history in the form of phylogenetic trees from that molecular data. That evolutionary history may or may not, depending on the context, resemble the trees that we get when we're looking at species. Okay, so that's what molecular evolution is. Why do it? What does it give us? Well, one thing, and we saw this with the 16S RNA, is that it gives us insights into early evolution, because not all organisms leave fossils, not all organisms leave macroscopic traces, but all organisms have DNA. And so we can look at the relationships of those organisms using DNA of genes that are conserved among all life, and we get ideas about early evolution, about how the very beginnings of the evolutionary process in life might have looked. The other thing is we get evolution among genes, not among species. So we've already seen that genes, for example, can be duplicated. We get paralogs. When we have speciation, we get orthologs. But when we get gene duplication or repurposing of genes from a gene duplication or repurposing of a domain from domain shuffling, we can see that evolution take place, and we can look at that evolution, and that is a process that's going on at the same time and leads to organismal and species evolution, but we're looking at it in a different way. So the other thing is we can also look, so in addition to just at the level of genes, how gene function arises and so on, we can also look at problems. We'll see, these can be kind of problems in the tree-based view of life, that is phylogenetics, and that is horizontal gene transfer. Maybe a gene gets introduced into an organism through, I don't know, a virus or some sort of infection or some other means. This happens in different species through different means. And when we look at the molecular level, we can start to unravel those types of complexities. Okay, so what does it mean? Okay, we've seen also already at the level of DNA sequences, at kind of looking at just individual sequences and sequence families, we've seen these processes, random mutations that get introduced and they get passed on to the next generation in DNA sequence terms. And we also see that favorable mutations accumulate and deleterious mutations are eliminated by selection. This is the two-step process of Darwinian natural selection. At the level of whole genomes, we have things like rearrangements and reversals and large-scale duplications and structural changes. But effectively, at the level of DNA sequences, these are the underlying processes. Now, we would like to be able to distinguish these, right? How can we infer the branching patterns themselves? That's the process of phylogenetics. So evolutionary trees, as we saw before, have a long history. They date back in civilization. Well, trees of life date back in civilization, thousands of years. Evolutionary trees in Darwin's thinking date back to 1837. And this is actually a figure. It's the only figure from The Origin of Species, and it's Darwin's attempt to draw a tree through multiple generations. We, like Darwin, are going to present these trees as branching patterns. So we start with a root and we branch out through the process of evolution. Now, it turns out that the pure branching pattern, in some cases may not be completely true, and in other cases it may not be completely known. But the branching pattern is a model that we use when we devise evolutionary trees in phylogenetics. And so we accept the limitations of this model and try to see when those limitations might apply. We're trying to get an accurate picture. So that means we want to get a tree that resembles as closely as possible the true structure of evolutionary history, so we can infer things about evolutionary history of genes or species from the trees that we get. This is done basically, it's a bioinformatics problem using the data that we get from, primarily now, from DNA sequences. Now, I want to just, before ending this short video, I want to raise an issue because we've already discussed paralogs and orthologs, and there is a complexity that we haven't covered, and this complexity gave rise to a new type of nomenclature that was proposed by Sonhammer and Kunin in 2002. And so in this case, let's say this is a tree from their paper, and we imagine some gene that is undergoing some speciation event. So this is basically the split of that tree into fungal and animal lineages. And then in the animal lineage, maybe there's a duplication that leads to paralog A and paralog B. Then we see a speciation. In this particular case, this is the lineage that leads towards worm and the lineage that leads towards human. And we see later more duplications. So what are the relationships of these? We can say certainly that HA1, HA2, and HA3 are paralogs. They're all duplicated, and we can see they're all duplicates of the ancestral gene HA. But what about HA1 and HB? Well, they're also paralogs, but they have a different relationship than, say, HA1 does to HA2, because this gene duplication dates all the way back to this point. So we can look at a particular duplication, and we can ask, did it occur before or after a particular speciation, assuming we know the branching order. If it occurred before, it's different than if it occurred after. So the HA1, HA2 relationship is different in some sense from the HB to HA1 relationship, because HA1 and HB split before the speciation event that led to humans and worms, whereas HA1 and HA2 split after that event. So this is the distinction that Sonhammer and Kunin make. They make a distinction between in paralogs, which are this kind of event, more recent than a speciation at issue. Out paralogs, which is this kind of event, that say the event that led to the A and B lineages, those are out paralogs with respect to this speciation event, and co-orthologs. So once we see in paralogs, we also see co-orthologs. So we can say that HB, we can say that WB and HB are orthologs, but this WA1 and WA2 are co-orthologs with HA1, HA2, and HA3, because they don't have the splitting that happened is all since that speciation. So in paralogs also create co-orthologous groups. And this type of more fine-grained distinction from just paralogs and orthologs is really useful when considering molecular evolution, and it's not something that we get if we're looking at just species evolution from morphological characteristics. So with that little caveat, that's where I want to leave this short video, and we'll move forward in this series of videos on phylogenetics.